Students, it is lecture number 2. In previous lecture, we saw what is the biotechnology and what are the basic definitions of biotechnology. Now we are going to see the first experiment of biotechnology. The first experiment of biotechnology in which recombinant DNA was found. That was done by two scientists, Stanley Cohen and Herbert in 1972. This experiment is also called Cohen and Bohr experiment. This was the first experiment in which a recombinant DNA was found. They isolated an antibiotic resistant gene from the plasmid of a bacteria. They used one another bacteria, that is the Salmonella typhimurium. They isolated the plasmid of Salmonella typhimurium. The antibiotic resistant gene and the plasmid of Salmonella typhimurium were attached with each other. For this purpose, they used restriction endonuclease enzymes. In lecture number 1, I told you what are restriction endonucleases. These are special type of endonucleases enzyme. They cut a DNA at a particular site. Students, remember one thing. Whenever we do recombinant DNA technology, whenever we do an experiment in recombinant DNA technology, and our purpose is to form a recombinant DNA, we use a specific restriction endonuclease enzyme. What they did, they used a restriction endonuclease enzyme first to have an antibiotic resistant gene, which was derived from the plasmid of a bacteria. Same restriction enzyme was used when they isolated the plasmid of Salmonella typhimurium for their experiment. With the help of same restriction endonucleases, they cut the plasmid of Salmonella typhimurium. It gets open like this. Now what happened? The antibiotic resistant gene and the plasmid combined with each other. They joined with each other to form a recombinant DNA. Now this recombinant DNA has a portion of plasmid, this one, and antibiotic resistant gene, this one. To join antibiotic resistant gene and plasmid, they used DNA ligase enzyme. DNA ligase enzyme. As we have seen in lecture number 1, that DNA ligase enzyme is required to join two DNA segments. And this formed a recombinant DNA. This was the first recombinant DNA formed in their experiment. And the next step was transformation. I have already told you in lecture number 1 what is the transformation. Transformation is the process in which a recombinant DNA or a DNA segment is transferred to a living cell. It may be a bacteria, it may be a plant cell, it may be an animal cell. And the next step was transformation. They entered their recombinant DNA, which they formed by joining antibiotic resistant gene and plasmid of Salmonella typhimurium into a bacteria, Escherichia coli. This Escherichia coli bacteria is just like Salmonella typhimurium. When their recombinant DNA entered inside the Escherichia coli bacteria like this, it started multiplying there. It created a number of copies of itself by using DNA polymerase enzyme of the host, that is Escherichia coli bacteria. And I have already told you, whenever a recombinant DNA multiply, divide, this is called gene cloning or the gene library. So what were the ultimate product of their experiment? What they did? They first isolated an antibiotic resistant gene from the plasmid of a bacteria, and then they isolated a plasmid from Salmonella typhimurium bacteria. Then they opened the plasmid with the help of same restriction enzyme which was used to have antibiotic resistant genes. Students, you can see the margin of antibiotic resistant gene. Look here, and also the margins of opened plasmid. Look here. These are called sticky ends. These are necessary to form a recombinant DNA. Why they are formed, how they are formed, that is a different story, and that we will see when we will study the detail of restriction enzymes. So what they did overall in their experiment? They followed all basic steps of genetic engineering. The first step was identification of DNA with the desirable genes, and that was the antibiotic resistant gene. The other step was introduction of the identified DNA into the host. That they did in the last of their experiment, where they transferred their recombinant DNA into Escherichia coli bacteria for gene cloning, for gene library. And the third step is maintenance of introduced DNA in the host and transfer of the DNA to its progeny. This happens automatically. Whenever a recombinant DNA is transferred into a bacteria, it stays here. It multiplies by using the DNA polymerase enzyme of the host and creates a number of copies. And when this bacteria will divide into two bacteria, the recombinant DNA will also divide automatically, so that each bacteria will receive one recombinant DNA each. Thank you.